Thank you very much, Dr. K, for that very elaborate and generous introduction. I appreciate it. So I want to talk to you today about the work that I've been doing for some time now. And as you may all know, alcohol consumption is a major risk factor as far as alcohol-associated liver disease progression is concerned. But recent findings have shown that there are several risk modifiers that could actually predict different pathogenesis scale or severity of the alcohol-associated liver disease. And so today I would want to walk you through how environmental pollution versus lifestyle interact as far as alcohol-associated liver disease progression is concerned. I will just walk you through a few things about the global context of non-communicable diseases, lifestyle, alcohol, and environmental risk. Also, I know Dr. K did tell a lot more about me. I will give a brief introduction about myself, and then delve deep into the actual research focus of looking at PFAS versus alcohol interactions. I'll walk you through some few key findings that we've been able to. So first off, our learning objective. First, I would want to help through this conversation describe PFAS and alcohol interactions. And by the end of this talk, we should be able to explain how PFAS as environmental pollutants interact with alcohol to affect liver disease. I would also touch on explaining the mechanistic pathways involved in this interaction. And then lastly, take you through some few translational implications modules that lie ahead on the way forward. So, as Dr. Cave introduced me earlier, I'm originally from Ghana, and I started my first degree in Kwame Nkrumah University of Science and Technology over there. Then, through recommendations of one of my mentors, then given that all the focus and attention at that time was just on infectious diseases, and how about the other aspect of diseases that are not caused by infections, but are caused by lifestyle or exposure, modules. And so that actually sparked my interest as far as reading toxicology is concerned. Went to Finland, University of Eastern Finland. And then from there, I was lucky enough to obtain the Japanese government scholarship to do a Ph.D. In Tokyo, at the Tokyo University of Science, where I worked with acrylamide, studying the role of NRF2 induced neurotoxicity. And after my PhD, had my first postdoc in the United States at the University of Tennessee Health Science Center, looking at nuclear receptor signaling and metabolic liver disease. And then, as Dr. Kim said, we met casually in a conference, American Association for the Study of Liver Disease conference at Washington, D.C. And fast forward, I was able to come into his lab to start my second postdoc, which led me into my faculty appointment from June of this year. So non-communicable diseases still do remain a significant challenge globally, and it's estimated that about 70% of death globally are attributed to non-communicable diseases. And even much more, the contribution of the environment is that much, about 23%. And as you look at the global map here, you can see the various places that are contributing to this effect. Normally, most of these non-communicable diseases have been associated with different types of lifestyle. And for example, the nutrition, physical activity, sleep or rest, alcohol consumption, smoking, and these together or individually in a way play several roles as far as associations with these non-communicable diseases are concerned. And so looking at the very root causes of the non-communicable diseases, as I did mention from the lifestyle, harmful use of alcohol and environmental exposures stand tall as one of the key root causes for these non-communicable diseases, which then bear their negative fruits of several non-communicable diseases. So let me talk to you about a little bit about alcohol and its epidemiological data so far. Consumption of alcohol still represents a very significant public health challenge globally. And this is leading to almost about 3 million deaths every year as a result of that. And Dali's alone is well above 130 million globally. And also here in the United States alone, excessive use of alcohol has been associated with about 95,000 deaths annually. All these are as a result of a spectrum of conditions or diseases or conditions referred to as alcohol-associated liver disease, which normally starts from simple buildup of fat in the liver, referred to as steatosis, and then further could progress to a steatohepatitis or fibrosis, which could further degenerate into cirrhosis, and then sometimes also could further on become carcinogenic to hepatocellular carcinoma. However, recent epidemiological and clinical data has shown that there are some variabilities as far as these ALD progression data are concerned. And the data shows that only about 35% of heavy alcohol users end up suffering from severe alcohol steatohepatitis. The 65% do not develop any serious illness, even though they consume this almost similar or they have similar patterns of drinking consumption of alcohol. This then establishes the fact that there are some contributing factors, for example, genetics, epigenetics, sex, gut microbiome, and for our discussion today and our interest, environmental exposures, which beyond the consumption of alcohol do play a role in determining the extent of severity for the alcohol-associated liver disease. And so we'll be looking at the environmental exposures in this context. This points out a very novel hypothesis in theory referred to as the two-hit hypothesis or multiple hit model. And this hypothesis essentially stipulates that liver disease progression is influenced by multiple insults. And so what happens is that there is a first hit which causes some minimal damage. And then in order for the liver disease to further degenerate or move onto a very much more severe condition, there should be a second hit which could be any of these risk modifiers, including environmental exposures. And so I would want to pause here and then just tell you a short story which I can use to introduce the environmental exposures and the concept of toxicology in perspective. So let's imagine a room filled with people, minding their own business, having fun and having their usual conversation. And then all of a sudden, a tiger invades the room. What is going to happen? People are going to shout, people are going to start running. Some might decide to go and give a call to 911 or pick up a weapon and try to, shut down the tiger or, do anything as much as possible to keep the tiger at bay. Now, in this room, there are different types of people: older adults, younger children, pregnant women, babies, nursing mothers, and people with existing health conditions, but to name a few. And all these are being faced with this threat of the tiger. And the fact that these different people have different strengths and have different levels of weaknesses will determine how they could easily become a prey to this tiger. So let's see how we can deal with the tiger, dealing with the tiger in the room. And this is relative to the usual mantra of dealing with the elephant in the room. But I felt that in order to help my message sink really down, let me use a model that is as ferocious as a tiger, which will get down to people. And so first, we need to identify the threat by asking some questions. What are the characteristics, features, how does this tiger attack? And that will constitute the hazard identification. Second, we need to understand the situation fully. How did the tiger actually enter the room? Was the room really locked and the tiger, broke it loose and entered, or the door was actually left open and the tiger could, these questions will help to provide very useful answers that will help in, keeping the tiger at bay in the long run. Which groups of people it prefers, what are the strengths and vulnerabilities within the room. These constitute the exposure assessments. Sorry. Third, we need to determine the level of risk. Is this really a threat to lives? I mean, compare the tiger versus, a cat invading a room. The responses to these two animals will not be the same. And so the question remains whether it's a real threat to life, what's actually the risk level, and should we be worried? And these will also help us, develop an immediate action to mitigate the effects. The fourth strategy is to develop control strategies by asking questions like, how do we keep it at bay? What control works best? And this constitutes the risk management. Now, the last bit is that not everybody in the city or in the country was at that point when the tiger invaded the room, located in the room. And so it's very necessary for news to go out there that, hey, there's a tiger around, there's a tiger in town. Everybody has to be careful, so that also constitutes the risk communication. So, lastly, as I did indicate, there are different people with different vulnerable, characteristics or features. And most of these are very sensitive. For example, young children, infants, pregnant women, elderly, chronically ill, workers who are always exposed at the occupational sites, and then the unborn babies. These are groups, we consider them as being very most sensitive to the attack of the tiger. Now, so what if I tell you that this story that I was, sharing with you was just a metaphor to establish the fact that the tiger that I alluded to represent actually the threat of chemical exposures that we face day in and out of our lives. And it follows the same trend where different types of people are encountered with different risk levels or different chemicals and environmental exposures. Humans are on a daily basis exposed to complex mixtures of chemical substances. These include from drinking water, breathing in air, consumption of food, and among others. In summary, for example, tobacco smoke, vehicle emissions, industrial emissions, aviation emissions, barbecue, food cooked over open flame, waste incineration, and then natural occurrences like volcano eruptions and forest fires. These make up just a few to cite as examples of the number of exposures that humans are encountered on a daily basis. Among these exposures, a unique class of chemicals referred to as the per- and polyfluoroalkyl substances, PFAS for short. And the PFASs have been grouped into several divisions, first as polymers or non-polymers, and then furthermore divided, if we take a look at the non-polymers, it's either that there is a polyfluoroalkyl alkylation or perfluoroalkylation. And then if we further zoom in on the perfluoroalkyl substances, that's where we have different compounds that have been also subcategorized into their N-term functional group. And so what happens here is that these PFASs over here to my top left, PFASs that have terminal carboxylic acid functional groups. And then these PFASs down here are the type of PFASs that have the terminal sulfonic acid functional groups. And depending on these different chemistries, they function differently and they could be used differently. It is worthy to note that given that PFASs have this fluorine-carbon bond, which in chemistry is regarded as one of the most strongest bonds, and that establishes the reason why they exist for so long in the environment without easily breaking down, and that's the name forever chemicals, which has been attached to them. So for the purposes of our conversation today, we would later on focus on one of the many PFASs, which is a sulfonic acid PFAS known as the perfluorooctane sulfonate. Uses of PFASs are numerous. Several personal care product, personal consumer product, as well as industrial product, industrial applications. And so, for example, non-stick cookwares, popcorn, microwave bags, and then fast food containers, to occupational use, for example, fire retardant foams, and among others. So let's zoom in on PFOS, perfluorooctane sulfonate, and then just look at a few characteristics that pertain to this compound. And most of these things are, very generic, related to all types of PFASs. They were produced and extensively used since the 1940s for several applications, firefighting foams, as you can see here on my top right, pesticides, flame retardants, among others. They remain a significant environmental public health challenge due to their widespread and the potential of bioaccumulation. And about 2 years ago, the International Agency for Research on Cancer classified PFOS as a group 2B, possibly carcinogenic to human compound. And the other legacy PFAS is perfluorooctanoic acid, which is the PFOA. During the same study session, they classified the PFOA as group 1, which is the carcinogenic to humans. And PFAS have been associated with several health effects, including mast LD, hepatotoxicity, immunotoxicity, among others. So why should we actually be worried about the PFAS story? If you look at the map here, you could see the widespread PFAS contamination that is ongoing in the country, and especially on the East Coast, where we have a lot of industrial activities having even much more higher level. But if you zoom in on the graph to my left here, you could see that Louisville is here right on top, within the top 10, having very significant amount of PFAS content. More so in recent times, research has revealed that wherever chemicals are present in beer, and researchers tested about 23 different beers produced around the country, and they found that out of the 23 beers, 22 of them have or had very high amount of PFAS that goes beyond the threshold that has been set by EPA. And most notably, the very highest amount of PFAS in these beers were found around beer producing factories that are located around the Cape Fear River Basin in North Carolina, which is an area which has a very unique history of PFAS pollution. And key PFAS found in these beers are PFOS and PFOA. And so this is a very worrying situation. So there is a critical knowledge gap in the sense that we do know a lot more about the harmful effects of alcohol consumption, and we do know a lot more about the effects of exposure to PFAS. However, it's still not very clear what the combined effects of alcohol and PFAS could yield to, and therefore establishes very huge and knowledge gaps. It also establishes limited understanding of the molecular pathways that underlie these interactions. There's also the lack of comprehensive toxicokinetics or dynamic data on PFOS in context of alcohol consumption. So the goal of our study was to explore investigations into the metabolic and disease-modifying effects of PFAS in context of alcohol. And we were looking at human alcoholic patients and then animal models. So first, we started the study of looking at humans, which was done in a cohort of DASH consortium cohort. And what we did was we de-identified plasmids from these cohorts from patients that have been categorized into either, having either severe alcohol steatohepatitis, moderate, and then we also had the healthy controls. And then we send out the samples. We process plasma from these, and then send out the samples to Emory, where we collaborated with our researchers over there, and they were able to conduct what they call the exposome analysis. And by exposome analysis, the exposome analysis is able to establish the overall number of exposures that these patients have been or have had over their lifetime, and using LCMS, GCMS techniques, we're able to establish the exposomic analysis as well as the metabolomic analysis. On the animal model end, we did what we call the reverse translational mouse model, using mice that were placed on alcohol first as ramp-ups, and then for 10 days, concurrent with 1 mg per kg PFOS exposure by oral gavage. And what we did was to divide these mice into 4 groups of either pair-fed with or without PFOS, or ethanol-fed with or without PFOS. So let me walk you through a few data from the human exposome analysis. First, we were able to find very interesting correlations between the levels of different types of PFASs and the severity of alcohol steatohepatitis. And so here you can see that PFOA was significantly correlated with severe alcohol steatohepatitis. And overall, we did see a very strong correlation consistent with all the different PFASs that were examined, albeit the PFOS p-value was not statistically significant, but we did see a clustering effect over there. And we also did metabolomics and pathway enrichment analysis, first in the plasma of PFOA established, exposed patients. And we did find several very interesting pathway changes, including the several amino acid changes, as well as glucose metabolic pathway changes. Then we also did that for PFOS, and then for alcohol steatohepatitis as well. And for PFOS, we did find very interesting terms related to vitamin C, pentose glucuronate, and then linoleate, as well as carnitine shuttle pathways. Same with the alcohol steatohepatitis. We also saw a lot of amino acid pathway changes, as well as bile acids, among others. So, walking you through the mouse model results, first we saw that PFOS exposure exacerbates the alcohol-induced steatosis and then liver injury. And this was evidenced by the level of alkaline aminotransferase, ALT, which is a marker of liver injury. AST was not significant, albeit there was a trend. And what we saw here was that whilst it's already an established concept in literature that alcohol consumption built up fat in the liver, with the exposure of PFOS, this amount of fat was even much more increased. Again, because we did see a lot of fat deposits in the liver, we sought to investigate or analyze lipids either within the liver or in the blood. And then we saw that there were consistent increase of plasma triglyceride and hepatic triglycerides, which also was consistent with plasma VLDL. The cholesterol, however, did show a general decrease in the plasma for alcohol consumption. However, in the liver, alcohol tend to increase the level of cholesterol, but with the exposure of PFOS, it went back to basal levels. And we also did see a general increase of non-esterified fatty acids by alcohol consumption, regardless of PFOS exposure. We also assessed metabolism of ethanol, and this is where we found very interesting results and what we call more or less like a paradoxical kind of situation. We did see that PFOS in concert, in conjunction with alcohol consumption, did significantly decrease the blood alcohol concentration levels and induced the messenger RNA upregulation of key ethanol metabolizing enzymes like CYP2E1, the alcohol dehydrogenases, and then the ALDH, which is known to help detoxify acetaldehyde. The reason why we refer to this data as more of a paradox is that we expected that with the enhancement of ethanol metabolism, it should have reflected in a very reduced level of toxicity or liver injury. However, we did see the opposite, and that's why we refer to this as a paradox. Because and also we were also able to corroborate the messenger RNA data with protein expression analysis, and then we did mass balance calculations, which essentially deals with looking at how much of PFOS was given into each mouse, how much of PFOS was stored or distributed across different tissues in the mouse, and then how much of PFOS was finally excreted or eliminated. When we did that, we realized that regardless of alcohol consumption, the majority of PFOS were stored in the liver, making up about 60% thereabout. And there were other organs which also had significantly high levels of PFOS also stored in them, but the liver had the most. Then we did transcriptome analysis, looking at different pathway changes for differentially expressed genes. And what we did find was very interesting. And so here is the heat map for, and what we did was to make different comparisons. And so this comparison here takes into account the pair-fed mice that were fed with only vehicle, and then mice versus mice that were fed with ethanol and vehicle. So pair-fed vehicle versus ethanol-fed vehicle. And then over here is the ethanol-fed vehicle versus the ethanol-fed PFOS. And in essence, this first group to the left establishes the ethanol effect, whereas this heat map to the right establishes the combined exposure effect. And so to actually bring or to establish actual relevance of interpreting the data from this heat map, what we did was that we did a further analysis that sought to establish the overlaps between the ethanol effects alone and then the combined exposure effect. And so here, we have the ethanol effect, and then here is the ethanol plus the PFOS effect. What we saw was very interesting and intriguing. First, genes like heme oxygenase 1, which is a target gene for the NRF2 pathway, were upregulated with ethanol alone. However, with the introduction of PFOS, this gene, which is a very good gene, important gene for the NRF2 pathway, leading towards antioxidant processes, was downregulated. Secondly, FGF21 was upregulated with ethanol, but downregulated with the combined exposure. And then CYP4A, 12A, and B, they are target genes for PPAR alpha. They were downregulated with ethanol alone, but with PFOS, they were upregulated, which is very consistent with data already existing in literature showing that PPAR, the different types of PFASs are key agonists for PPAR alpha. And then lastly, we saw commonalities between the two, that CYP2C55 and 2B13, which are target genes of CAR, were all upregulated regardless of the different exposure treatment. Same as the CYP3A59, which is a target gene for PXR. And then looking at some pathway enrichment analysis, we looked at gene ontology, KGG pathway, and then we also used the Ingenuity pathway analysis software tool to analyze. What we did see were pathways that were related to lipid, fatty acid, metabolic processes running through consistently. We also did prediction of the long-term effect of this exposure, which is done by the Ingenuity pathway analysis software, and it predicted that conditions of liver carcinoma, liver cancer, and hepatic steatosis, which actually we had already seen in our morphological data, as well as fibrosis of liver. And then we also did some qPCR analysis of key nuclear receptors and their respective target genes. And so we did look at PPAR alpha and its target gene CYP4A10. We looked at PPAR gamma and its target gene CD36. And we did look at PXR and its target gene CYP3A11, as well as CAR and its target gene CYP2B10. And interestingly, they were all significantly upregulated by PFOS with alcohol combination, apart from the PPAR alpha itself, which did not see much change. Lastly, we also did what we call the upstream regulator analysis. And this analysis was supposed to tell us what types of different transcription factors could mediate the alcohol PFAS or PFOS interactions. What we found was that whereas there were several transcription factors identified, PPAR alpha came as tops as a transcription factor that was key in mediating the effect of alcohol plus PFOS combination. Very interesting is that we did some side-by-side comparison of the human data and the mouse data. And so far, it's very refreshing to note that there are some overlaps. And we saw that looking at the pathway analysis from the mice and looking at the metabolome-wide analysis from the human samples, linoleic acid metabolic processes were common, between the mice studies and the human studies, which is very refreshing, because then it reassures us of how translational our I'm sorry, our data could be towards human relevance. So in summary, from the mouse data, we were able to establish the fact that combined exposure of PFOS and alcohol, number one, increases buildup of fat in the liver. Second, it has a decreasing trend on body weight. It also increases liver to body weight ratio. And as I did talk about the paradoxical situation of having significantly decreased blood alcohol concentration levels, and then increased triglyceride consistent along with VLDL, increased VLDL levels. And then lastly, as a marker of liver injury, ALT also increasing. There were also enriched hepatic transcriptional pathways, which were associated with lipid metabolism, steatosis, oxidative stress, and xenobiotism. So, few conclusions here. First, these studies put together provides for us novel insights into the potential interactions between environmental pollutants and lifestyle factors. Again, it also establishes the fact that PFOS exposure may increase the risk of promoting ALD. And then also, there is also the potential for environmental pollutant to exacerbate the detrimental effects of alcohol consumption. And then lastly, these findings highlight the need for targeted interventions, which will be aimed at mitigating the adverse effect from these combined exposures. So, few ongoing studies and future directions. Given the data I showed you about PPAR alpha emerging as the top transcription factor, which is predicted to mediate the alcohol PFOS interactions, we wanted to explore further to establish the exact role of PPAR alpha as far as the PFOS and alcohol interactions are concerned. And so we're going to we're using animal models like PPAR alpha knockout mice to establish this. And also, just as I did talk about from the overlap data comparison that we made between the human data and the mice data, we want to further evaluate how relevant our data could be as far as human relevance is concerned. And so we are using a humanized model of PPAR alpha mice to also establish, to establish the same exposure models. And then also, we also wanted to expand the scope of the different types of PFASs that we have examined. Because, for example, in this first study, we did work with PFOS, which is a sulfonic acid type of PFAS. But we wanted to also look at how about carboxylic acid functional group PFASs. Do they also share the same common pathways as the sulfonic acid functional groups, or they do have different pathways? And how about the new next generational PFASs or the alternative PFASs that have been produced? So that research is also ongoing. And then also, yes, we need to find solutions to mitigate this effect. And so through a collaboration with our colleagues from Boston University, we are trying to see how we can use dietary fibers, which our colleague in Boston University has shown that giving dietary fiber and PFAS at the same time resulted in the PFAS being bounded by the dietary fibers and processed for excretion in feces. And so we want to explore more research into this and see how this could be a very key intervention or mitigating effect for the combined PFAS or PFAS alcohol exposures. And then lastly, we're also expanding the human exposome analysis by including more research subjects, and then also expand the scope of PFAS. We want to study even delve deep into different types of PFASs that are related as far as these human cohorts are concerned. So recently, our paper got published, this work got published in Toxicological Sciences, and UofL News picked it up for a news release. And so I would entreat you to scan those codes and then, read those, either the paper or the news item. And following the news item, as Dr. K did mention earlier, we were invited for an in-studio interview at the Kentucky Education Television, where we were able to, explain in very fundamental terms the relevance of our study. Lastly, also, this table here shows that we are only dealing with PFASs, but up to date, there are about, let's say, about 5 different studies that have considered other types of environmental pollutant. For example, 1,2-dichloroethane, ambient particulate matter, the Gulf War illness related chemicals, polychlorinated biphenyl, and then with PCB 126. And so about four different compounds that have shown key changes or key mechanistic findings as far as environmental pollution with alcohol combination is concerned. For the first time in many years, the Gallup polls predicted that alcohol consumption is on a decreasing trend. And I asked myself, could research like what we are doing have, could it have played a role in this generalized, decreasing trend? But I believe that it's very satisfying to see such kind of trends going down. So in closing, I would want to remind us about my tiger metaphor, that PFASs remain a significant environmental targets that we face every day. And we need to stay alert, remain vigilant, and work together to tame it. And so I want to end this talk by saying a few words of thanks to my mentors, Dr. McLean, who is my division chief, Dr. Cave, and then my lab personnel, Adrian, the Cave lab, and my other collaborators, and more so, very especially to say a big thank you to the Louisville Clinical and Translational Research Center for selecting me as a presidential research scholar. And these are photos that we took from the in-studio interview with KET Network. And I must admit, I was a bit nervous because that was my first time having cameras all over me. Lastly, my first, which I consider very humbling, my first and official assignment, having assumed office as a faculty, was to work with a high school student, Inas Saad, and I was able to help her appreciate the concept of science, said that she emailed me a few weeks back that she really wants to come back every now and then, and even sometimes, communicate with me on several experiments that are done in their schools. And I felt that that was a very good start for my faculty journey. And so to this extent, I would pause here. Thank you very much for your attention, and also kindly solicit for your comments, questions, or discussions.
Comments, questions, Dr. Ekuban, Dr. Mercer?
Yes, sir.
Very nice presentation.
Thank you.
You've done a lot of very nice work, and I think it's going to lead to a lot of attention and better understanding of these chemicals. You focused kind of, so you looked at the metabolome, and then you looked at the transcriptome, and you looked at proteins, and you highlighted certain transcription factors that tend to bind these types of molecules. Are you working with anybody or considering those particular transcription factors and modeling in the PFAS, PFOA, those different chemicals to see if there is a expected affinity and a range of affinities?
Yes, thank you very much. So we currently are preparing an R01 together with collaborators from Boston University and University of Massachusetts Lowell, and that is one of the specific aims that we are looking at the exact role of PPAR alpha and how it connects to different types of PFASs. So, yes, yes, sir.
Looks like in your animal studies, you use male mice only.
Yes.
Is there anything known about sex-dependent responses to PFAS?
Yes, thank you so much. I mean, this was one of the key critiques we had during the stage of peer review. And we did explain that given that this PFAS with alcohol combination model is very novel, we wanted to start with an animal model that is well experimented and well studied, so that we can have fundamental or foundational mechanisms that could easily be expanded on towards the future. So it's within our immediate future plans to look at females as far as starting with PFOS is concerned, and even expanding it beyond that.